Hi, my name is Cindy Chespick, and I'm an associate professor of biomedical engineering at the University of Michigan. And I'm going to be your moderator for this session, which is entitled Developing and Distributing Novel Electrode Technologies. So you're going to hear from a range of groups that have each developed a novel tool enabled by the Brain Initiative for electrophysiology. And each of them are going to talk about, the unique capabilities of their tool, and then also some of the challenges of distributing those to a broad group of neuroscientists, or planning some of that distribution. So throughout the talks, we're going to be monitoring for questions, and at the end, we will have a live question and answer session. So we're looking forward to having a conversation at the end of this. So without further ado, we will go ahead and get started with the presentations.
So today I'm going to talk to you about carbon fiber arrays for high-density neural interfaces. And so we've been very fortunate to be funded by the Brain Initiative to both develop and distribute these tools to neuroscience labs. So the core mission of my lab is motor neuroprosthetics. So what that means is that we're trying to take signals from the nerve and the brain and use them to control multi-degree of freedom finger movements. And while there's really a lot of things that you can accomplish with a Utah array, ultimately we're not going to have enough. And so that made me very interested very early on in carbon fiber arrays. And that was based on the work of T.K. Kozai and Tim Gardner in showing that you could use a 8-micron carbon fiber, so an electrode smaller than a neuron, in order to record in the brain. So in my lab, we turned this into an array of individual carbon fibers. So what you're seeing there is sort of our older work from 2015. These 8-micron carbon fibers are about 150 microns apart. And, due to the sort of strength of carbon, if you can have 500 to 750 microns of free length, and you can still just drive that into the brain despite their 8-micron size. So you're seeing that in a rat brain on the right, and what you're seeing is it's partially carbon fiber, and there's also a little bit of help coming from mechanical silicon supports. So over the last few years, we've developed and used a wide variety of these devices. So on the left is our older workhorse, where we were first getting these into the brain by dissolving PEG as they inserted. You can also get pretty much anywhere in the brain with a cannula, and then have carbon fiber for the last 500 microns or so. But obviously, the PEG is a pain. The cannula is definitely undesirable. So we've been developing, and I think are finally in what we can call beta testing for devices that can sort of go anywhere in a rat brain. So what you're seeing here is a device where there's, at the very end, it's all carbon fiber. And then there's, you can see these gradations going back, and that is gradually widening silicon to support the length of that carbon fiber. And there's a channel inside of the silicon that holds the carbon fiber. And we have 3 millimeter, 6 millimeter, and 9 millimeter versions of this. We did a lot of alpha testing, trying to get them to insert with as little material as possible. And this is sort of, the final dimensions we ended up with. And we can attach this to any kind of connector. And you're looking at 16-channel arrays here. So there's a lot of really cool electrodes, and you're going to hear about a lot of them in this symposium. So why might you as a neuroscientist want to use carbon fibers? And I think that it comes down to, if you need the mechanical properties of the carbon fiber, or if you need the chemical properties of the carbon fiber. So in addition to being, much better able to penetrate tissue, it's also surprisingly robust to work with. So this is, an early version of our nerve device, and it is embedded in silicone, which helps. But you can see we can run this over, these are carbon fibers, with a glass capillary 10,000 times. And this same test does not work for silicon, for example. So what this means in practice is it can handle more surgical handling. Also, carbon fibers are very commonly used in neuroscience for sensing dopamine, and this is a large part of the development effort that we've been doing, and right now we're in the process of debugging new rigs in the Becker, Burke, and Garris labs for 16-channel fast-scan cyclic voltammetry using these new devices. Also, I'll say, it's not just about the quantity of channels, it's also about the quality. And so I'll show you histology in a second, but these electrodes are particularly non-damaging. And my student Alyssa Welly spent a lot of time sort of engineering the tip to the point where we can get a very low impedance electrode with still a very small cross section for recording. And that has sort of enabled us to get to the point where we see, good units on basically every channel. And this was just published in the Journal of Neural Engineering. And I'm showing you acute on the top, chronic on the bottom. And this work has sort of led me to think, okay, if you're not seeing a unit at least 100 microns or 100 microvolts or more, there's probably something wrong with the brain or your electrode. Another thing that this is particularly good for is, again, when you have something very small you want to record from that is nonetheless sort of hard to penetrate. So our collaborators, Tim Bruns and Ahmad Jaman, just uploaded this paper to bioRxiv using carbon fibers to record from rat vagus nerve. And so that's a 200 to 300 micron structure. You have to penetrate epineurium. And we've been really pleased with the signal-to-noise ratios that we can get on these carbon fiber electrodes. And even on the lower left there, you can see what appears to be tracking and action potential across 4 different electrodes on our 16-channel array. Another good capability of these carbon fibers is that we believe we can pretty closely identify the location of the tip, or at least we can identify the tip of the hole that the electrode was in. So this is, based on 2D images. My student circled 3D images. I made a 3D movie all the way down, and the green are neurons. And again, we know that these electrodes land very close to the neurons. So I'm showing data on the right from a 13-week implant. And, we and others have published histology on carbon fibers, but it's really cool every time to see similar neuron density on the top and on the bottom. So these are not statistically significantly different in, the number of neurons in these images. And you're seeing half the array here, where, we are able to sort of, advance the image until the black hole disappears and see where the tip of the electrode is. And, these are action potentials that we recorded from day 84 on these electrodes. So, we have every channel has a closest neuron and a biggest waveform. We're not deluded enough to think it's going to be that simple, so it's going to take a lot of work to sort of show, whether or not we can actually associate individual neurons with their waveforms. But the high density of neurons is really consistent with, seeing more single units that you can sort on these electrodes. So, in conclusion, if you need a robustly inserting electrode, or you want to localize it precisely, or you want to record dopamine, I think this is a really good technology. So in terms of distribution, we've had the good fortune to be able to distribute a lot of devices through the NeuroNext program. So Yusik Yoon runs the Mint Hub at University of Michigan, where anybody can Google us, go to our website. If you click carbon microthreads, it brings up a form that goes right to my excellent full-time technician, Juliana Ritchie, who then, contacts the labs and we figure out, what they need for their experiment. And through this program, we've been able to distribute 345 devices to 33 labs. And so among those 33 labs, I think only one of them wanted the actual picture that was in our data sheet. And we were really surprised. Everybody else has wanted some degree of customization for their application. And it is a benefit of, having a few, adjustable by hand steps that we're able to accommodate a lot of these requests. I do think for distribution, this would be really challenging for a company. And so, to talk about the cost of manufacturing this, from a non-company academic lab, we actually did a detailed cost study within our own group of what it costs us to make these, sort of anticipating, what we're going to do after the end of some of these programs. And let me start by saying, there is a remaining by hand step, which is the laying of carbon fibers. This is a video from my collaborator's lab, actually showing it being done not by hand. So there's a robot, an extruder, micron stage, and you actually see a carbon fiber being laid down on the pad. Play that one more time, and it actually gets sort of sucked into the silicon channel during fabrication. So the materials and clean room cost to us is $160 a device. If you add in, all of the time of people doing the fabrication, that comes out to $350. We kind of like that number. And then they made us include management, overhead, administration, and then it becomes $600 a device to sort of what it costs as a whole at the University of Michigan. So in practice, we're sort of, writing small bits of grants for, a few $1,000s worth of devices. But we're trying to figure out a way to make that sustainable to, our existing collaborators. Though I think this is, we're primarily distributing, in addition to NeuroNext, via, collaborations where we, as the engineers, go into a small service role. We're very proud to help Jill Becker get preliminary data towards an R01 on social support and addiction, which she's going to use the dopamine sensing capabilities of the arrays. We're also really excited to participate in Galit Taleb's new project, where we're going to use the penetration capabilities of the arrays to penetrate some octopus epineureum. Apparently, the tentacles have a lot of neurons in them, so we're very excited about this project as well. And we're sort of continuing to sort of do, small in-service collaborations to try to get these out to, the biggest Electrode users that have been identified through, our NeuroNext and through our internal projects. So with that, I just want to thank my lab. The people that do the most work on the carbon fibers are Parath Patel, Alyssa Welly, Joseph Lettner, and Juliana Ritchie. And I'd be happy, please enter your questions in the box, and I'd be happy to answer them by email as well. Next up, we have Chongji from Rice University.
Thanks, Cindy, for the introduction. I'm Chongji from Rice University, the Department of Electrical and Computer Engineering and Biology. So today, I'm going to talk to you about our effort on the ultra-flexible electrodes. So this is an effort we have been working on for the past 6 years. Thanks to the organizer for this opportunity to present our work here. All right. So here is a very simple short outline. So since we're in the relatively earlier stage than most of other speakers in this session, so I'll emphasize more on the current capabilities and our effort of this also flexible electrodes we have been working on. And later on, I'll touch upon the effort we just initiated to disseminate these electrodes to other labs. So we are among the believers that electrodes need to be flexible to become friendly to the surrounding tissue. So we took a step further and make them also flexible. And I'll show you how we make them stably record from individual neurons for many months, and I'll also show you how we scale things up to the scale of thousands of neurons. All right, here are a few examples of our electrodes. As you can see in these figures, that our electrodes are very similar in terms of the design from any other passive recording electrodes. So the only difference we make here is that we make them from polymers and we make them extremely thin. The overall thickness, as you can see, if we measure across such a probe, is just a micron. And the rationale we're running here is that the flexibility of such a device is determined by the bending stiffness of the structure. And the bending stiffness, if you look at the equation, is proportional to the Young's modulus and also T to the cube to the third of thickness. So that means if we switch the material of the probe from rigid materials such as silicon and metal to polymers, we gain a lot in terms of flexibility. But a more effective modulator here is the thickness of such a device. If we make a device from 10 micron in thickness, which was the typical thickness back when we started this project. If we make it down to 1 micron, we gain 10 times in thickness, but we gain 1,000 times in flexibility. So given this principle, we were able to engineer the flexibility, engineer the device to maximize the flexibility of these devices. So we call them nano-electronic thread, or in short, NET. So with this, we were able to make extremely flexible devices. Here is an example of such a device. This is a 128-channel device suspended in water. These devices do not self-stand in air. They will just collapse. But if you suspend in water and drop a cup of water nearby, and you can see that they are driven crazy due to the extreme flexibility. If you were to measure them quantitatively, here we have an example measuring the bending force of a suspended device with the atomic force microscopy, so that you can see the bending force is actually in single-digit nanonewton range. Just for your information, this single-digit nanonewton is within the similar range of a single cell's traction force. So again, this is due to the super thin structure of such a device. All right, with such a flexible device, the difference we can make is once we implant such a device, here you can see the two-photon image acquired while the implant is still in the living animal. You can see an implanted electrode as shown as the red ribbon here. It's getting along with the surrounding capillary very well with no BBB leakage at all. On the right, you can see that there is no observed neuronal degeneration happening over a course of months during implantation. All this data are published a couple of years. I refer you to this paper for more details. So far, I've shown you the evidence that these devices get along with the surrounding neuron tissue very well. You may wonder that whether they report at all, because we make the electrodes so thin that it's highly questionable whether they can maintain their electrical functionality at all. So back then, there were a lot of questions, but it turns out they can record just as nicely as any other passive recording electrodes. Here is a few examples showing how you record and the typical units isolated with from this recording. And the real difference we can make here is that we can actually turn this recording to be very, very stable. So the stable here has two meanings. One is short-term stability, and the other is long-term stability. So let me unfold here a little bit more. So in the long term, because the electrodes get along with the surrounding tissue very well, we can maintain a relatively stable performance for those electrodes over a time course of months or up to a year. If you look at the measurements of the recording performance of those electrodes, the impedance and the unit yield and the recorded accident potential amplitudes are relatively stable over the course up to a year. So far, we have tested many of those devices, and in most cases, we can yield a stability over 6 months or up to a year. And the failure often comes from connectors, just as we expected. And we have tested these electrodes in mice, in rats. We just started to test them in non-human primates as well. We have tested brain structures such as cortex, and started to use them for deeper regions such as hippocampus. So this is about the long-term stability. And in the short term, many of us know that if due to the mechanical mismatch between the implanted electrodes and the surrounding soft tissue, even in short term, there are relative motion happening between the electrodes with respect to the tissue. So you don't get to record from individual neurons very stably. You often lose them after a few hours. Here is an example of our electrodes. On the left, you see a 128 array. And on the right, you see the isolated unit out of such array. Each dot here represents the estimated location of one of those units during one recording session. Now, if we come back a day or two after, you would expect another set of recording. But whether you can match those individual dots and conclude we are recording from the same neuron, so it's hard to say. Let me show you what it actually looks like when we actually do that. Now let's zoom into one of those dots. That means we look at the recording of this one single unit. If we unfold this piece of data and present the waveforms recorded by the neighboring channels surrounding this neuron, you see there are 4x8 waveforms showing this, showing the recording from the same neuron. That's from the neighboring channels surrounding this neuron. Now that's the first piece of data. All right, if we were to play this video here, each frame here is showing you a piece of data from 1 hour of recording. And this recording was continuous for over 2 months. And now I'll just fast scroll this video because it takes a very long time to show you the whole picture. If I go all the way from beginning and to the end, you can see that there is really little change in the waveforms. That's because it didn't change at all, very much at all. So I hope this piece of data can convince you that this, first of all, this Electrode didn't drift or move much over the time course of almost 2 months. And the recording was pretty stable and was stable enough for us to patch up all the data and continuously track the same neuron over 2 months. Now let's look at how we can actually scale this type of recording up to a few thousands. So here is an example of a piece of 128 channel of NET device. And over the past years, we have put in significant effort to diversify the designs of the contact arrangement and geometry of those contacts. And we have concluded to be a standard design of those devices, and we turn them all into 128 channel modules. And we practiced a lot of surgery and got a standard practice to implant many of those modules in one animal. As you can see on the right, here is an example of 8 of those devices implanted in one area with a spacing in the cortex with a spacing of just 150 microns. So with this capability, we can achieve very high density recording using these electrodes. Here is an example of 8 of those 128 channel devices, as you can see, covering an area in the V1 region of a rat. And this dot array is showing you the anticipated implantation location of each contact of those 1024 in total channels. And this is a micro CT data showing you where they really end up being. As you can see, they all squeeze into this tiny volume about just 1 microliter. And here is an example of our recorded data. Just like a typical passive electrode array, we can record local field potential in this region, and we can isolate all neurons firing all the spikes that's being shown in this movie. And to conclude, we have all the data summarized, and this plot is showing you all the units we sorted out. There were over 1,000 units recorded by this 1024 channel array within this about 1 millimeter cube of brain tissue. With this data, we were able to play a lot more interesting games. For example, we can look at the connectivity between units, just as an example. So with this dense sampling, we can cover a significant region of one region. For example, in this case, we had 10 of those 128 arrays covering a significant portion of B1 and higher order visual regions in the mouse. And this video is showing the electrode coverage and the local field potential and all the unit activities. And given the same electrodes, we can try to distribute them across the cortex as well. In this case, we have 20 of those devices covering a variety of various cortical regions. Again, the electrodes and local field potential and unit activities in this experiment. So with that, I hope that I shown you that this ultra-flexible electrodes can help us stably record for a prolonged time duration, and we can possibly scale them up to record thousands of neurons at a time. Now let's talk about how difficult it is, or what different it is here to really send these electrodes to other labs compared to conventional approaches. And I'll also discuss about the challenges here. So there are mainly two difficulties in terms of sending the electrodes to other labs. First, we needed to develop a specialized assembly and implantation strategy in order to implant those ultra-flexible devices. As you saw, these devices are extremely flexible. They won't go into the brain by themselves. We need to design a shuttle device. In this case, we simply use the array of tungsten wires and pre-assemble those flexible shanks onto those wires glued together by APEC polyethylene glycol. And when we lower the assembly into the brain and we retract the shuttle wire array and leave the probes inside. So in terms of dissemination, we would need to pre-assemble everything, which is manual work, assemble everything before we ship them to our collaborating labs. Our collaborators would simply need to implant them using a standard stereotactic technique. So the other difficulty was a specialized backend. In this effort, we collaborate with a company called SpyGadget, and we also took an existing working strategy developed in Loren Frank's group. And using this set of head stages, this set of stackable head stages, we can scale things up to a few thousand, up to 3,000 channels in a behaving animal. So with that, I would like to discuss future challenges. One of those challenges we're trying to address is to standardize the fabrication and also standardize the assembly procedure, which is currently manual and pretty laborious. And we're currently in the SBIR project working with SpyGadget to improve along this trend. And the other difficulty was to really surgically implant so many devices and also securely fix the head stage onto the head of the animal. And the surgery like this typically takes 8 to 9 hours in our lab. And if we could turn this bulky head stage into some ASIC chips binded on those devices, it will simplify things a lot, very much. So we're collaborating with a few groups to work on this trend as well. So with that, I'd like to thank you for your attention and appreciate all my lab members working on this project, especially my student and current postdoc, Zheng Zhao. He did most of the work I presented here. And also my collaborators, Len in Rice as well, and Loren Frank in UCSF, and Matthias Carson in SpyGadget. And thank you for all the funding support from Panat and Wells. Thank you for your attention. And our next speaker is Jim Harris from Genelia Farm.
Thank you. Today I would like to tell you 2 short stories about 2 generations of electrophysiology probes that were developed by my group at HHMI Genelia Research Campus. The first locally and the second through an international consortium of donors. So the first one is about conventional silicon probes, and I think it's a useful lesson for people who live in academic environments, because we operated most of this within an academic environment like technology base. It was entirely driven by users. I am not an electrophysiologist, and I do not use silicon probes for my own work. So shortly after I got to Genelia in 2009, I had people in my office saying, there are these things called silicon probes, but they don't have enough sites on a shank, they don't have enough sites on a probe, and we need to put more than one probe on an animal in order to push forward. This is 2009, so at the time, a 16-channel probe was an exotic piece of hardware. And so I responded to that by looking at the available technology, then sold by NeuroNexus, developed at the University of Michigan, and thought, well, I think that available fabrication technology could do a bit better. The technology from NeuroNexus at that time was late 90s capability, and there had been some commercial advancements that would make it better. So I thought, all right, we can do up to 64-channel probes and maybe 16-site shanks. It would be better if we could do more. And the problem with more channels on a probe is that if you were going to use these in freely moving mice, the cables were unwieldy. There were no multiplexed head stages in 2009. But a little tiny company called Intent Technologies was selling a 16-channel multiplexer chip that we thought could be used. The trouble was it didn't work with silicon probes, and so we bought 160 of them so they could change a design feature of it so that the low frequency cutoff was shifted, and that's how we built the first available multiplexer head stages. The probe development, however, is somewhat less happy. We first tried with a commercial fab in California called IMT, and we tried to replicate the NeuroNexus process. And unfortunately, one of the critical materials that they needed was not available, and they absolutely and completely failed to develop it. We spent more than $250,000 and walked away absolutely empty-handed. However, later, Satiris Masmonitis used that same foundry, and they succeeded in getting good probes by using only processes that were in-house and perfected. The second trial was at the UC Berkeley fab, where that material was available. The difficulty is that we spent 70% of our money calibrating tools and only 30% of our money actually making things. We got probes, but it was really painful. And so the lesson was, in that case, that you can do it, but it's really hard to manufacture something if you want to manufacture it reproducibly in a teaching fab. It can be done, but you really have to spend a lot of money. Teaching fabs are there to teach. I don't resent that fact. I just want to, make people aware that manufacturing in that environment is really a struggle. And so what we finally ended up doing is moving to the NIST fab, which has a program that external contractors, especially if they're working for nonprofits like HHMI, can use their capability. We changed the design radically to something we thought was simpler. We got a lot of probes. The lessons we got from that is life is better if you have a great process engineer. And we had a young lady named Andrea Price who was just spectacularly good at trying something, failing, trying something, failing, and trying a third thing and succeeding and having nothing but good news for her weekly reports. I think we could have learned more from experts. We tried pretty hard, but we didn't learn enough. And if I had to do this cycle over again, I would try harder. And I think we were a little cautious. We should have tried hard stuff sooner. Go ahead and make what you want to really make. And if it doesn't work, back off instead of the other way around. But you need someone committed to selling probes if you're going to finish this. And that's one of the issues. The world doesn't need another probe paper. It needed probes. So the issue was lithography, is that the development of the original probes was done with quite modest lithography. We stuck with that modest lithography quite a long time and finally decided to use something called projection lithography, which was in 2008 available in almost no university clean room. It is now available in most of them. It came online in NIST about the time that we thought, let's go ahead and do this right. And so I ended this project with making a probe with a 64-channel shank. It was only 78 microns wide, which fit my criteria. You could have gotten 64-channel shank probes before, but they were so wide they were not stable in a chronic implant in a rodent. And then another probe with two 32-channel sites. This is by far the most popular probe that came out of this. You can buy these things now from this little company called Cambridge Neurotech. And so my lesson in this is dissemination has to be committed at the very beginning. If you're not committed to disseminating on day one, you just shouldn't do this unless you absolutely, for your own science, need a new probe. You need someone fully committed to selling something. That was not me. And luckily, Tal Holtzman came along, and he spent 2 years of 12-hour days creating a market, and these are now successful probes that are used around the world. The second story I want to tell you about is about NeuroPixels. These are CMOS programmable probes, and there were two problems that we left with. One is I didn't want to wire out more than 64 channels, and I couldn't make more than 64 channels on a probe that I could make, and so you needed to go to a professional fabrication facility. So we needed better lithography. You can get 384 channels on a shank. If you can do better lithography than you can do at NIST. The second thing is that you needed to integrate amplifiers and multiplexers because you don't want to wire out a 384 channel cable off a mouse. And so you need fully capable CMOS from design, fabrication, assembly, and testing. Testing costs as much money as fabrication does. You need a manufacturing commitment on day one. That was when I went looking for somebody to do this. My first condition was, if you don't make them and sell them, I'm not going to give you any money. And you need someone that's committed to selling them. That's not so easy. You know, I don't know how to collect sales tax in Brazil. And the third thing is you need a support plan. Who's going to answer the phone if somebody calls, and what does that do to the price and the breadth of impact? And so my recipe for this is, look, write a spec. Even if you're going to make something yourself, you should write a spec. What must it do? And then ask yourself, does anyone but me care? It's great. It's great fun to make a thing in a fab. It's almost never worth it if no one else needs it and you don't need it to do your own science. And then the question, is it worth it from a dollar standpoint? Real CMOS design and delivery costs several $1 million to engineer and a couple of 3 years. And then you need to produce something like about $1 million worth of material to maintain a device in a modern fab. If you're not going to go through that, you're going to become obsolete quickly. And I want to emphasize again that the fabrication of the chip is never most of the cost. It's tied up in all the other steps of making something. And so just very briefly, NeuroPixels got over all of those humps, not all without trials and failures. In 2019, they delivered 3,230 probes to more than 230 labs. There are now those numbers are over 4,000. They're sold at cost, 1,200 euros each. Support is free, meaning it is provided to people who do it out of the goodness of their hearts, mostly by my group at Genelia, but other founder labs have been generous in their time of answering questions and helping people. The two lower pictures, the lady on the left is Heidi Henning, the lady on the right is Chris Heiberger. They are the lawyers at HHMI that made these contracts. Without them, this would never have happened. So what's going on now is that we are testing a new probe, which is a third the size, and so you can put two on a mouse, 768 channels, selectable among 10,000 sites, weighs less than a gram. You'll be able to buy these in about 1.5 years. And the NIH has generously agreed to fund what I think is the end of the line, what I call terminal technology, a probe that you can see on the left there that we call NXT, has a tiny base. It has no head stage. We think that it will allow you to record brain-wide, limited only by the number of shanks you're willing to insert in the brain. You generate an immense amount of data that's challenging to transmit out, but it's a solvable problem. The world knows how to do high-speed data transmission. And so in conclusion, completion requires a commitment to manufacturing and distribution. That costs time, it costs money. There's opportunity cost. If you're a graduate student or a postdoc, this is not what you're supposed to be doing. You need a plan as to who's going to do that, who's not trying to either get a PhD or finish their postdoc. Thank you, and now I'm going to turn the ball over to Allison Yorita.
Hi, everyone. Thank you for being here today. My name is Allison Yorita, and I work at Lawrence Livermore National Laboratory as a staff engineer within the Center for Bioengineering and the Center for Micro and Nanotechnology. And today I'll be giving a really brief overview of the work that we've done underneath the BRAIN initiative focused on the distribution of flexible polymer microelectrode arrays for multi-species applications. So to start off with, I wanted to go into what we're actually distributing. So at Livermore, we focus on creating flexible polymer microelectrode arrays for electrophysiological recordings. An example of this is seen on the upper left-hand side in that picture. So that white block you see on the right is a connector that leads out to the electronics, and on the other end, you see those tiny little forks. Those are the areas that actually go into the brain, and at the very tip of those are the microelectrode arrays. So far, we've created channel counts ranging from 32 to 128 channels. I say so far because we're actually working on higher channel count devices as well, but I'll go into that later on. At Livermore, we can process these using a robust process technology. And so we use standard MEMS fabrication techniques to make all of our devices. And what's great about our facilities is that we actually have tools specifically set aside to fabricate these devices. And so we can do all of our fabrication under ISO 13485 quality control procedures. In addition, this is a platform technology, so it can be expandable. And we've created devices ranging from animal to human use, although today I'll be focusing specifically on animal use devices. And so the benefits to our polymer probes are that they are thin and flexible. And related to this, our collaborators have seen long lifetimes. So our main collaborator and the head PI of this project is Loren at UCSF, and his group has seen lifetimes of up to 250 days when implanted in rats. In addition, they've demonstrated large-scale recordings as well as single-unit stability. And so to that, I want to show this plot here, which was published in 2019 out of Loren's group. And so on the left-hand side of that y-axis, you see the number of units measured per electrode. On the right-hand side, you see the number of units measured per shank, and this is a 16-channel shank. And so with this graph, you can see that his group was able to track and monitor single units using our devices over at least 160 days. And so using this technology, we were really excited and encouraged by this result. And so we wanted to go ahead and try and distribute these and get these into more people's hands. And so I wanted to walk through the steps that we take to actually get these devices out the door. And so the first step involves the microfabrication of these devices. Again, this is done in the clean room at Livermore. And so the first step involves taking a substrate wafer, so silicon wafer, and depositing a layer of polyimide on top. So that's that yellow layer. The next step is to deposit a thin film metal. So this will define all of the trace metal as well as the electrode metal. We can then pattern the metal using standard lithographic techniques. We can then deposit another layer of polymer. This will act as the insulation layer. And finally, we can pattern that polymer and expose the electrode as well as define the outline of the device itself. And then we can peel the device off of the substrate wafer, and we now have a fully flexible microelectrode array. And so the next step after these come out of the clean room is to assemble them. And so the first step is to connectorize the device. So you can see in that blue circle, that's where we add in this example, a 36-pin Omnetics connector, although we can do other types of connectorization as well. The next step after that is to then electroplate. So these devices, when they come out of the clean room, have platinum microelectrode surfaces. And so we can actually modify that surface using electroplating to make it more sensitive to picking up electrophysiological signals. And so in these two examples here, we can show that we can plate PDOT on the left-hand side and platinum iridium on the right-hand side. And so the PDOT layer is actually that really dark layer that you see over the top row of electrodes, and the bottom is the bare platinum. And then on the right-hand side, the platinum iridium are the darker gray films that you see, and then you can see three bare platinum electrodes interspersed throughout as contrast. And so finally, the next step is to prepare these devices for surgery. And so again, because these devices are fully polymer, they can't actually go into the brain on their own. And so we have to devise and fabricate these silicon stiffeners to help them get into the brain. And so that's what you see in box A. And so that's a silicon stiffener. You can see that there is a channel etched down the middle of the shank. And so the first step is that we apply what's called here a glue. So that's basically polyethylene glycol, or PEG. And so we apply a little bit at the very top. When we melt the PEG, that'll wick down the middle of the shank, down the entire length. We can then flip that over, which is seen in box C, and attach it to the probe. So the PEG will adhere the probe to the stiffener. And in box D, you see a fully attached device. And so what this actually looks like under a microscope is seen here at the very bottom. So this is a 2-shank device. You can see that it's been plated with PDOT. And so the probe is on top, and then underneath that, you can actually see the silicon stiffener peeking out. And so finally, this is an example of a fully assembled device that's ready to ship. And so this particular device was a practice device. It's made fully of polyimide only. It's not used for actual recording. It's what we send out to collaborators so that they can practice and refine their surgical technique. And so you can see at the very top, that's a 3D printed dummy omnetics. And then at the very bottom is a silicon stiffener. And then through the middle, that T is actually a polyimide wing, which aids in the surgery. And so once these actually get into people's hands, I'm going to walk through how they do the surgery. So you can see here, they attach the probe to a surgical arm, which is this gray arm sticking down. I've also labeled the connector, the wing, the probe, and the stiffener. And so to start off with, this is inserted into the brain. The next step is to build up a well around the insertion site. So this can be done either using dental cement or a custom 3D printed piece. Those polyimide wings are then attached to the well for stability. Next, the connector is moved away from the surgical arm. Saline is then applied to dissolve the PEG that's holding the probe to the stiffener. And then finally, the stiffener is fully removed, so it's attached to the surgical arm, and the probe is still in the brain and ready to record. And so next, I wanted to go into the structure for how we actually distribute these devices under this project. And so after we fabricate and assemble everything at Livermore, we then send all of the devices to the lead lab at UCSF. So Loren Frank's group is responsible for the distribution, surgical training, and troubleshooting advice. So the first round of devices go to what we're calling the core labs here. So these are labs that are located at UCSF, UC Berkeley, and University of Washington. And they're responsible for the surgical development, surgical training, device testing, and feedback. And so we have lead labs that focus on rat, songbird, mouse, and non-human primates. So once they've refined the surgical technique for each species model, they can then teach this surgery to the support labs, which you see on the bottom here. And so these labs will also receive devices from UCSF, and they're responsible for the device testing as well as providing feedback. So this feedback will go up to the core labs, up to UCSF, and then ultimately back up to us so that we can further make design modifications. And so I wanted to walk through this structure that we set up for this iterative redesign of the polymer probes. So the first step involves the device design for different animal models. And so this is done at Livermore using feedback that's provided by UCSF. The next step is that labs then pick which designs will suit their animal model or their studies. And so again, I mentioned there's that blank practice probe that I talked about a couple slides ago. And so for the first pass, we distribute these blank practice probes to groups to refine their surgical process. After that, they then receive the actual microelectrode arrays. So the next step, once they pick their design, is that we then can fabricate and distribute these probes. So again, we fabricate and assemble at Livermore. We then send all of these probes to UCSF for further distribution. And finally, the participating groups will implant and provide feedback on the designs. And again, because this is an iterative process, we want to take into account all of their feedback so that it can feed the next round of cycle fabrication and distribution. And so I've been speaking mainly so far about the distribution of these 32 to 128 channel count probes. But what I also want to emphasize is a really exciting thing that we're doing in this program is also kind of pushing the limits of what we're able to do on the probe side to create an even higher channel count probe. And so right now we're actively working on creating a 512 channel count probe, and this involves 4 different entities. So at Livermore, we're responsible for the polymer probe design and probe microfabrication. Lawrence Berkeley National Lab is responsible for the integrated circuit design as well as the fabrication for the CMOS chip. Spike Gadgets is a company based in San Francisco, and they're responsible for designing the channel acquisition system. And finally, UCSF is responsible for the probe distribution, system integration, and software development for this probe. And so because we all need to work together to ultimately create one final probe that can be distributed to everybody, there's a lot of kind of these relationships at play, right? I can't just make one decision and assume it will not affect anybody else. And so to make sure that we all stay on the same page and that we maintain communication with each other, we have periodic in-person meetings. It does help that we are all somewhat located in the San Francisco Bay Area to do this. But when we can't do that, which for example now, we also utilize Zoom, Slack, Dropbox, and Smartsheet to stay on the same page. And so finally, to wrap up, I want to emphasize that at Livermore, because we do have these quality control practices, we found that this is absolutely key to producing consistent and reliable probes. And finally, funneling of the right information to the right parties is very important. So whether that's information on how to do the surgeries flowing from the core labs to the support labs, or whether that's feedback from all of the labs, making sure that it ultimately makes its way back up to us so that we can make modifications to our designs for the next round of probes, all of these control of information is important. And finally, I wanted to talk about a few challenges that we found from this distribution project that perhaps we can talk more about in the Q&A. So the first being that a lot of the participating labs have a significant hurdle of actually being able to test the devices when they don't actually have the funding to do the testing. Secondly, the refinement of these designs does appear to be more species dependent than anticipated. So for example, probes that we thought would work for mouse and rat, we thought might work for songbird, but we are finding that might be actually not the case. And so we're looking at ways that we can refine some of the dimensions to make it so that they can work for songbird. And finally, many steps in the post-fabrication assembly are actually done serially. So once they're done, taken out of the clean room, we actually assemble and connectorize those devices one by one. And so my group is looking at new ways that some of these steps can be done in a batch process. And so with that, I'd like to thank you so much for your time. I'd like to thank my team members at Livermore, as well as my collaborators in this project, which include Rosie Huck, which is my co-PI at Livermore. Loren Frank is the lead PI of this project at UCSF. Peter Dennis at Lawrence Berkeley National Lab. Beth Buffalo at University of Washington. And Matthias and Magnus Carlson at Spike Gadgets. And so with that, I'd like to wrap up my slides, and I'll be happy to introduce Eric Itri at Carnegie Mellon University. Thank you.
Thank you for the introduction, and thank you all for joining us. My name is Eric Itri, and this work, together with Rahul Panat and our teams listed here, as well as Mason Chamanzar's group, is about a fully customizable 3D printed electrode array. And the real purpose of this array is to address the challenges that we experience when we use electrodes. So in a given experiment, say we want to hit hippocampus, striatum, and prefrontal cortices in a mouse, this presents many issues. This is a target. This is a multiple targets. We need a customized setup. And ideally, at least for several of us, we would like to do this in a freely moving animal. Surface microelectrode arrays won't be able to hit these deeper areas. And to hit this with multiple silicon arrays or tetrodes, this would include neuropixels, would be costly, difficult, though potentially doable, though not so easy to do in a freely moving animal. Similarly, more and more people are looking to incorporate electrophysiology with imaging. And so we may want to put electrodes around the outside or even in the middle of a window. And achieving this would be very difficult to do. So together, we've developed an array called the CMU Array, which is a 3D nano-printed array that is capable of 6,400 channels, and that's one shank per channel per square centimeter. And we can place these channels anywhere we want to with high density and on any substrate. Actually, everything that you see here, including the circuit going out, was 3D printed. So this allows an incredible amount of flexibility. And the shanks are very narrow, making, even with this high density of array, makes it very easy to penetrate the brain. Just as a sense of the full customizability of these arrays, we can print our university's name, CMU. Here on the right, on panel G, that's a 512 channel array. And also part of this is that we can not only get these dense and intricate layouts, we can make the tips pointed. It also allows us to route the signals out to any connector that we need. So if you're using an Omnetics connector, we can use that. Samtec or a ZIF, that's easy as well. We're just 3D printing the leads in a fully customizable way. But one issue we had with this density, as well as this wanting to have flexibility, is how to actually achieve this layout without a printed chip. And so in order to do that, we actually, as I mentioned before, are printing the entire chip, and we're doing it in a multi-level procedure. So we are printing pads for all of our electrodes that can be 1 millimeter high, it can be 4 millimeters high, any length that we want. And we're printing out on every layer one set of leads, then covering it with Parylene C, then printing another and another as we see as we go left to right. And then on top of this, we can print the actual shanks that we want at the depth and location that we desire. And then this whole chip can be implanted into the brain either acutely or chronically. In terms of how well it goes in, here's just an example that I showed you before of that 100 shanks being implanted into the brain. We dip this probe first in dye, leaving behind these nice little marks. And one of the impressive things that 3D printing in these very narrow shanks has enabled is that we no longer have a need for specialized equipment for introducing these electrodes into the brain. This probe was lowered into the brain just with your benchtop stereotax. There's no need to shoot it in or vibrate it in, and the damage it causes is actually quite minimum here. If you look at this histology slide and try and figure out where exactly the shanks were, here's where the shanks were, and it's really difficult to see any amount of tracing. This was an acute prep. Shanks were lowered in for several minutes, dozens of minutes, and then pulled out. But the real proof in the pudding is the quality of signal. And so unlike silicon arrays, with the spacing that we have here, we're only getting 1 or 2 neurons per channel. And here's 32 channels. We're seeing now most of these channels, we're getting good isolation of neurons. In this case, we're able to easily identify just with envelope thresholding where those 2 neurons are. And so we've developed a very flexible array that will enable anyone to easily and quickly, printing one of these arrays only takes a handful of hours, get a specific targeted electrode, and fairly inexpensively, that we can use in any animal and in any circumstance that you need. But 3D printing isn't limited to just this avenue. We've actually been looking into delivery, rather than just recording, but delivery of fluid or light. And so these electrode shanks can actually be made hollow, opening up the possibility of fluid delivery or potentially in the future microdialysis. We haven't looked at that so much, but allowing us to puff on a bit of drug through the middle of these electrodes and then look at the local and long distance effects of these of the administering of this drug. Similarly, we can also print polymer that can act as a waveguide. Here we have it bending around a corner, and what this really enables, which I think is pretty fascinating, is that if you have an experiment, say you want to look at functional connectivity throughout this area of brain. In vivo, in a awake animal, you can devise, as you will, an electrode to sample throughout that 3D printed volume, and intermixed on this electrode will be recording sites as well as waveguides, which would enable you to then, hope you can see the blue dots here, flash through, look at the responses, both locally and long distance. You can do these stimulations paired in one area or across different areas, targeting the inputs for whatever your recording is. So this really is a phenomenal amount of flexibility that this project has enabled. Although this project is actually quite young, and I think one of the great things that the organizers did here is to get together, one, the great ideas, but also projects that are at many levels of development. And so we've really seen, in terms of how we've been progressing forward, first and foremost, a coordination between engineers and biologists is very, very, very important. It's never great to see people who build a hammer and then go out desperately looking for a nail. And so to have this alignment with either the biological or clinical needs is fundamental to early support and early success for the technology. Also looking ahead at packaging, not just the electrode itself, but packaging at a larger scaled up point of view. Related to that also are the challenges that we have for scaling up generally, the techniques that might be able to make 1 or 5 or 20 probes are different from 500 or 1,000 probes. And thankfully for this process, like I said, we can print several hundred shanks in just a matter of hours, but we're still looking ahead to the challenges that proper scaling up will have. And then last but not least, always seek out resources early and often for tech transfer, because the real success of a technology isn't that it can do something, it's having people adopt that technology and use it. And for that, you typically have to go well beyond just your lab or your set of groups. So that's an overview of the CMU Array, some of the challenges that we've faced and our trajectory moving forward. I'd like to open this now up to the live conversation and questions.
I think we may have, Cindy may be having some technical problems. So until she comes back on, I think we may as well go ahead and get started. Cindy, I apologize for usurping your moderator role, but I felt like we should go. Well, once you're back, I will step back. So my name is Loren Frank. Oh, there you are, great.
Hi, sorry about that. Sorry, I thought that I was going to be auto unmuted, but I was wrong. Thank you, everybody. So let me go ahead and get started. I apologize for that. So Chong, there were a lot of questions during your talk about insertion that I think you got to, but are there concerns about breaking the electrodes when you insert? Like, is that part of why these surgeries take so long, or what is the most challenging thing about these surgeries?
Cindy, thanks for the question. Again, it's for the pro-Christian type or it will stay long, I guess. But despite any, regardless type of the be careful to take on those pro if you're careful trained as out of the earlier find the first
Sean, we're having some trouble hearing you. I think that
not at all. Just volume.
Okay, maybe let's go on to another question, and we can sort of see if support can help with your audio. Let me ask a question to Tim. So Tim, at what level of electrode distribution, either in terms of a quantity of devices or a number of labs supported, does it make sense to transition to a company? And can you sort of put a number on that?
Yeah, I mean, I guess just within my own experience, so we have 3 levels of experience at that. One is supply to Genelia labs. And so I wouldn't want to supply any significant number of labs, more than just a handful, unless I had a paid technician. It is not appropriate work for graduate students and postdocs. And so if you're going to supply even half a dozen labs, which is what I was doing at Genelia, I wouldn't do that with people who are trainees by the definition of the NIH. I just don't think it's fair to them, and it doesn't really advance their careers. But you can, with a single technician, you can certainly supply 10 or 20 labs, maybe even 30. I think going beyond that is getting to be challenging, but it can be done. And then if you so the experience that I had with transitioning to production was, it depends on the market, is that if you're going to sell things for a living and you're going to do it for your primary job, I think you need to be thinking in terms of many hundred probes and many tens of labs, preferably hundreds of labs. And it depends on what the per lab buy is. That Neural Pixels is pretty rigid. Their minimum order is 6,000 euros worth of probes. And yet they still, are approaching 300 labs worldwide with more than 4,000 probes delivered. That's probably not a typical example for something launched out of an academic group, because it takes a lot of legwork to get to that level. And so I think that the simple answer to your question is, think many tens of thousands of dollars worth of devices sold before you can even consider a part-time company, and hundreds of thousands of dollars sold if you have permanent employees per year.
Okay, thank you very much. So let me sort of go through maybe some of these technical questions first. We can talk about the electrodes, and then we can talk a little bit more about the distribution. We have a number of questions here about also the 3D printed probes. Eric or Rahul Panat, can you maybe tell us about the different materials that you can use in your 3D printing, both for the electrode and also for the insulation of these devices?
Yeah, so I can talk about the materials. So currently we started with silver. That was the easiest available nanoparticle ink that we could use with the aerosol jet printer. So the first attempt was to create these silver shanks and then electroplate gold on top of them. The second one is we are currently able to print gold shanks as well, right? But they have their own. The process needs to tweak to each materials, obviously. So we are able to print gold, but the next steps will be to look at platinum and platinum rhodium, which would be again, we will have to tweak the process, but we believe that this can be done. Yeah. So currently at this time for metals, it is silver and gold, and for insulation materials, typically we print polyimide, but we are also able to print PDMS. As an insulation material, which would give us a choice of multiple polymers for insulating these metal ones. Yeah.
Very cool. And I think I saw a slew of similar materials questions come in for Allison as well. So do you want to, can you talk a little bit about this sort of range of materials? It was very cool how you were talking about having sort of dedicated equipment, but what sort of the range of materials that you can use with some of your designs?
Sure. So especially with regard to the clean room tools that are set aside for the fabrication of these probes, there's actually a very short list, especially with regards to the type of metal. So for example, we can deposit platinum, iridium, gold, amongst a few other metals. But there is a very short list, and we try and keep that very controlled because we do have the capability of putting these into human, which is really great about our facility. But I guess related to that does mean that there's less or fewer materials that perhaps that we can kind of use to explore. And luckily, because the clean room is not just consists of our tools, there are also other tools that are more for general use for other groups as well. And we do have access to some of those other tools to do kind of more exploratory research. But at the same time, I think you do lose a little bit on some of that ability to do that quality control throughout the process.
Fair enough. Okay, Sean, are you with us now? Hello? Can you try speaking for a second? I can try again on the question.
But there weren't.
No. Unfortunately, no. So, at some point. All right, so maybe let's start talking about some of the questions about distribution. And so here's a question. So, let me pose this sort of broadly. Probably all of you are working with labs that are going to be the initial alpha testers of your technology. What is the funding model? Because it takes an awful lot of work for a new lab to bring up a new technology. Do you think that this should only be done with full-time personnel? And for example, and who should that personnel be? Should it be a technician, a grad student, a postdoc? Like, what's the right model for that? And maybe I'll ask Loren first, since I saw you nodding your head.
Yeah, Cindy, it's a really great point. I think this is one of the challenges we've had is we getting probes to other labs is great, but having them have the personnel to actually test the probes and give us feedback when, for example, they may not be funded to do so is a really big ask. And so I think, for this sort of thing, they're really, we're fortunate to have some funded labs, but for the others, it's their goodwill and their time. And so I think it's not a great model for actually doing the kind of distributed testing. And so, I think, for example, what's done with NeuroPixels has been spectacular, right? But Tim has had this amazing sort of team, I think, of people and himself dedicated to this. And most of the rest of us don't have that. So I think from my perspective, the idea of having grants that really provide money for the distribution and testing is a critical phase of this, and then moving it to companies as quickly as possible for anything that looks like it has potential. But it's a hard road.
Actually, maybe then I'll ask Tim next. So are NeuroPixels so great that people can get them up and running for free? Or at the beginning of this, was it sort of harder to get them up and running in labs around the country?
So Cindy, NeuroPixels is a really bad test case for NIH-funded work because we had partners with deep pockets to start with. Genelia is historically sort of an unfairly deep-pocketed, and so we just committed my group's very large budget and the large budgets of other labs to do the testing, and write the software. And the Allen Institute was similarly posed. I mean, they had deep pockets and they had their own internal budgets. The other partner was UCL, or the Sainsbury Wellcome Center, through John O'Keefe's lab, and we got a grant from the Wellcome Trust on their behalf, 1.5 million pounds. 900,000 of those pounds were used to pay for fab, and 600,000 pounds were used to pay for test. And John complained bitterly throughout the entire 4 years that he was losing money on every test. And so I think from the standpoint of just, listening to how much does it really cost? It's hard to keep the real test and design and feedback budget below the fab budget. It's kind of a 50-50 deal. And so now that I've written, some NIH grants to support that, that's not a bad guess, is that if it's going to cost you in the fab to deliver devices for test, $1 million, you better have $1 million budget to feed the academic side of it, to actually do the real testing and to interface with the engineering design groups all along, because it's an iterative process. You write a spec, they go to work, they find out, gee whiz, that's really hard and it's really expensive. Do you really need to stick to that spec? Or how about, can we back up a little on that spec? Or on the other side, we made mistakes and said, look, we just forgot something that was really important. We got to have it. What can we trade? We're willing to relax other specs if you give us that capability. And so those people have to get paid. They have to pay tuition, all that jazz. And so I think that a good guess is 50-50. If you think, you're spending $300 to make a device just in fab costs, you should have another $300 to pay the people who are going to use it. And that doesn't count assembly, Cindy. The devices that we get are assembled by our fabricators. It's once they're assembled and ready to go, you need that much money again to just make sure that they are ready to go out the door.
So I'll just add, I think in terms of personnel effort, we're sort of in agreement. Yeah, it's about a 50-50 as how much work it is to sort of get something up and running. Eric and Rahul, is that similar to your experience about the distribution of effort between the very first users and the fabricators?
Yeah, that is what we've seen so far. And I mean, we are both on this Brain Initiative grant together and split it down the middle. So yeah, in terms of the effort and the cost, it's absolutely true. And then one sort of caveat underneath all this that I want to point out is, particularly if you are early days, it is so very important to have a nail already and be building a hammer, rather than I think I can build some fun electrodes and have no biologist actually in need of that. And I think that's largely reflected in what all of us have been talking about. But too often I've seen engineers trying to go it alone, whereas the biologists are really the end goal. So don't discount them.
Yeah, I think I totally second what Eric just mentioned. Just being able to build a device, right? This was a cool technology that we were able to build, and using this has a lot of fluid dynamics to it. But at the end of the day, when we are building a probe, right, with different shank lengths, different materials, and other things such as the hollow shanks that Eric just showed, it really boils down to what is needed to study specific biological questions, right? What is useful to biology is what we need to focus on, and that's what I'm happy to mention. We are both of us are doing that.
Okay. All right, Sean, we're going to try you one more time. Do you want to try again? And let's.
Yeah, can you hear me now?
Question of insertion of your devices.
Can you hear me now this time?
Yes, I can hear you now.
Okay, great,
yeah.
Perfect. Thank you for hanging on there. So I guess there was a question regarding whether these probes are resilient enough during insertion and implantation. I guess they are indeed very dedicated, but if we can avoid applying big forces on them, which we do for any type of probes, they're all fine. So as an example, if we pull the electrodes into the tissue, they are fine. We never had failures with that. If we pull the electrode out of the brain tissues, they typically can survive that as well. We never had them break. So if we can avoid bumping the probes into anything, so which if you're careful doesn't happen, so they're usually fine. I guess that answers the question.
Sure. And just because we didn't have you before during some of these other questions, have how has your experience been with translating some of your probes to other labs? Are you having challenges in terms of how much time it takes for that other lab to get up and running, for example?
All right, thanks for the opportunity. I do want to apologize to my collaborators. Thank you very much for your enthusiasm, but I do want to apologize if you haven't received our probes as I promised. I said over the question system that I'm very cheap because I promised to many labs. But in reality, like Tim has mentioned, that it's very difficult to have students who are on training for their main goal in my lab. So it's hard to force them to support other labs without any scientific output. So with that said, it's been a long procedure, and we had some progress, but it's definitely not efficient enough or as I expected. So as I mentioned, we are trying to explore the way to commercially deliver these probes to other labs, but in a way supported by federal grant. And we hope to ramp this up so that it can be standing alone and self-sufficient for the rest of its lab. So everyone who are trying to get our probes or any probes from this session, stay tuned. I think everyone is working super hard to deliver them.
So I want to talk a little bit more. I see some questions here about the commercialization. But first, I want to go back to Allison. You said something during your talk about national labs being in a particularly good position, to do this. What do you foresee as the sort of role of Livermore and national labs in distributing devices? What kind of technologies? Is it things that aren't quite, commercializable or too advanced to be easily commercializable? What is the role of national labs?
That's a really good question, Cindy, and it's actually a discussion we've had a lot within our group at Livermore as well, because we do think of ourselves as kind of right in between maybe academia and industry. And so while we're not allowed to, actually sell devices and distribute them in that sense, we can really only do it through grants. But I think what's great is that we are able to kind of do some of these more slightly larger scale fabrications of perhaps more novel technologies for kind of this mid-level distribution. But I mean, one thing that we have discussed is whether or not we can move some of our, more basic devices to a fab, for example, that could enable other labs to kind of use our technology and get them up to speed, so that they can kind of jump into kind of these more, as we kind of try and push these technologies even further, whether that's, adding different modalities to the probes themselves, expanding beyond EFIS, or whether that's, even higher channel counts. So that's kind of where I see the national labs as being in part of all this distribution.
Sure. So now maybe let's talk a little bit about commercialization. We have a question here. Is there room in the market for all these probes, or do the panelists expect that the market will consolidate on a few of these designs? Would these groups plan to collaborate to encourage that consolidation? I don't know who to pick on here first. I guess I see Loren nodding again. But
excellent. I think it's a good question. I guess I would make the point that different needs drive different devices. So it is my guess, although I would love to be wrong about this, that the unit stability that you can get from flexible devices will never be matched by silicon. In that you can record for many days, but the units move up and down. That's the classic observation as far as I understand, which means that these devices are suited for different things. On the other hand, the density of CMOS sort of things is harder to get in the flexible devices. So I can imagine that a set of devices will survive, but I think it's actually going to be probably less which ones are best and more which ones have a decent business plan. Right? Like NeuroPixels will not go anywhere, I think, thanks to all the work of Tim and his colleagues to make this incredibly robust foundation. I'm hoping that the polymer probes also have that. It'd be great to see the 3D printed probes have that similarly, but setting that up in terms of the foundation of a distribution pipeline is tough. And I'll just add, as part of the work, we're collaborating with Chong and Lan on distribution of their probes, one of the things that we proposed is indeed a test across different devices to directly put them in the same animals at the same time to ask the question, can we really say, oh, look, these devices are good for this, these other devices are good for this other thing? And I think that would create market space for multiple devices, but it's still to be done.
Yeah.
Sure.
One comment I have on the same question is the way I look at it, and I come from a microelectronics industry. I was at Intel for about a decade. So when it's like in pathfinding phase, right? We are in the research phase when we are looking at different technologies, but some of the technologies that have already matured, they will dominate for about 5 to 10 years before being replaced or modified by the newer ones that come in. So it might be like a sawtooth type of trajectory rather than just saying that a few of the technologies that will kind of hold true forever, because newer advances will always be coming, right, from multiple labs.
Sure.
Cindy, let me add something since there's a different perspective, I think, from me. First of all, I'd like to push back on Loren is that there are many publications of 6 months of stability for silicon probes cemented to skulls in rodents. So you just don't need flexible technology to achieve that outcome.
But not of the same neurons.
However, I think that remains to be proven. I would argue there's no possibility we aren't recording from the same neurons, but they do move. And so you're going to have to do something to prove that you've got the same ones. I think that can be done. I think much more importantly is that we have seen many reports from high quality users that CA1 hippocampal neurons are not stable across many months with silicon probes, either NeuroPixels or others. I don't understand why. You could have accused the old silicon probes of being too short an array and it moved. You can't accuse that of NeuroPixels. You can't get lost. They just go away. We've seen the same thing in cerebellum with fewer reports, with the same users getting, just month after month after month of stability in cortex. The thing that puzzles me now, Loren, is that the people who are using them in hippocampus say, I just can't keep them more than a month or 6 weeks, so plan your experiments accordingly, are loving them in entorhinal cortex, which is posterior and deep. And so why you can get such great recordings over time in entorhinal cortex when you can't get them in CA1 puzzles me. But what I'm telling people is, look, the silicon probes just don't seem to be working in places that you should try something else.
So yeah, we could actually spend a lot of time here. It would actually be really, I think, fun for our audience to get us all talking about the relative pros and cons of these different devices. I personally think there's room for a lot, and it's a sort of per experiment sort of thing. But I wanted to get to this other question here, which I thought was really interesting from Nick Steinmetz. For some of the probes that have extremely difficult insertions in surgeries, and I can also imagine for probes that have very difficult rigs setups, where it's just sort of really hard to stand up a rig. Are there any thoughts towards distributing data sets rather than distributing probes, perhaps some sort of observatory model? So have any of you thought about, whether the right thing to do is to build one amazing rig, that does your thing as best as possible and try to, farm out time on that rig for data sets? I'll open this up to everybody.
Hi, Cindy, can I say something about that? Oh, you can hear me. Okay, great. I guess that's a great model, and no one actually has focused on in the past. But indeed, I have thought about this. I talked to Loren about this since years ago. I think this is a great idea, especially if coming to this. Nowadays everyone is trying to record from thousands, or if not hundreds of thousands of neurons at a time. This data set and the recording rig, and the cost is enormous. And the time cost and the economical cost of doing this becomes really, really prohibiting. And sorting the data, everything becomes prohibitive for individual labs to do so. And animals are not, plus surgical technique is difficult to implant many probes in one animal. But if we can come up with a unified model or a scheme, we can do experiment. We can totally do this experiment in our lab and post the data somewhere, probably online, and post all the parameters and experimental setup, everything, so that everyone can analyze the same data. It will be great. I totally like this idea.
Yeah.
Okay.
Yeah. Just
let's see.
Maybe
Eric. Oh, there. Eric or Rahul.
Yeah. Yeah. Okay. Just one comment on the same topic is as any standardization is going to lower the barrier for entry for many labs. And as my discussion with Eric has made it clear, cost as well as lack of standardization are the two major obstacles in expanding the market, right? So I don't know the number, but maybe let's assume 100 or 200,000 probes are used per year. To take it to 1 million or 2 million, I think the cost needs to come down significantly, and so is standardization that is needed. So this is absolutely a very good thing to have.
Okay, so we're coming up on 1 o'clock here. So I'm just going to ask one last question, and maybe we can do this by show of hands. People want to know and asking questions, are these probes available, and should they contact you if they are interested in using your technology for these experiments? So maybe by show of hands. Okay. All right. And you guys, I think there's many different stages, but yeah, so I think everybody's email address is we're all very Googleable. And so I think there's hopefully a lot of interest that we've been able to drum up during this session. We really want to see these technologies make their way into more people's hands over the next few years. I think that's the end. So thank you very much for attending.
Thank you, Cindy.
Sure.
Thank you.
Thanks a lot.
Thank you.